one level above that in the atomistic world, looking at biomolecular systems. And so he received his education, gets up to master's at University of Moscow. In Moscow.
It's Moscow Institute of Physics and Technology.
Okay,
it's different places.
Okay. And then received his PhD in biomedical engineering from Boston. And so it's a delight to have him here.
Yeah, thank you very much for the introduction. And yeah, I guess I will show you that we're going like scale up, but actually some of the math which we are using actually does come from quantum mechanics. So actually as you know, science and math and physics is very tightly interconnected. So people find something in one area, it turns out to be it's very effective for the others. So hope it will be like an interesting story. So okay, so essentially the main work which I'm working is on modeling macromolecular interactions, so specifically protein-protein interactions, and this is kind of and one thing is first, before you start, why this is an important problem, and there are several reasons for it, right? So essentially, from one thing, proteins are major building blocks of all cellular activity. So whatever kind of you do, in the end, if you want to understand cellular mechanism, you need to know how proteins interact, right? And at the same time, I mean, we were very successful in sequencing in getting protein sequences, right? That information how the proteins built up, right? And that was this human genome initiative, right? I mean, we had a tremendous progress in sequencing in the last 20, 30 years, right? But interestingly, with the structural methods that we did develop in the last 40, 50 methods, we developed methods for structure determination like X-ray crystallography, NMR, and others, but they actually turned out to be good for identification of structure of individual proteins. But if you're interested in the complexes, the rate of growth of complexes is very, very small. So it's really kind of it's a challenging problem, and there is no progress actually. NIH, they had a big kind of protein structure initiative, and they paid for last 5-7 years, they paid a lot of money to crystallographers to solve this problem, to do the same thing for sequencing, just to get new structures, but this didn't happen. I mean, so individual structure, yes, please, but complexes, it's very difficult to catch them because they kind of come and leave, so it's very difficult to deal with them, right? So that's why computation actually is very important for such areas, right? Because I mean, experimentally we are kind of we are lagging, and there is no way actually to do progress. And finally, why protein-protein interactions are important, right? That it's actually very interesting in terms of if you're interested in drug design, right? For targeting some particular interactions that you really want, you need to know, you need to understand the structure, how it works, that you can actually disrupt it with small molecules. So that's why this problem is important. And so let's just kind of start. I mean, as I presume, kind of most of the audience is not really doing that much of biology, right? And honestly, myself, I'm kind of more a physicist and mathematician. So let's kind of start with the problem formulation, when we can cast, right? I mean, the biology to some kind of math and physics problem, and then we can actually look how we can solve this type of problem, right? So the first and simplest problem which we deal with, I mean, is we want to deal with is called so-called docking problem. Essentially, finding a structure, when you're given structures of two separately determined proteins based on their crystal structures to determine a complex, right? So why this is important, as we discussed, I mean, you can easily get individual structures, but not the complexes. So in this formulation, right, I mean, we in principle can, if we can solve this well, I mean, that actually can be a big progress for the biology field. So from physics viewpoint, right, what we are dealing with, right? So we are having a statistical, it's in principle, it's a statistical mechanics or statistical thermodynamics problem, right, where essentially you have a tremendously huge configurational landscape of all possible protein kind of configurations, right? I mean, and you want to find a global minimum of free energy of this system, right? So it's kind of easily formulated, but as you know, right, I mean, proteins are it's a molecules which consist of a huge amount of atoms, right? So we're having a huge configurational spaces. So unless we do some approximations and simplifications, it would be difficult to solve this problem. And actually here is it's kind of like what is kind of like my area of work is actually trying to come up with such type of formulations where we can actually solve this problem one way or another and actually get some meaningful answer, right? So as I told you, right, even if we deal like essentially we consider just two proteins with limited flexibility associating together, right, we had a huge configurational space. So we have like billions of potential configurations how two proteins come together. Additionally, like as I told you, like in addition to kind of this, many proteins don't change too much conformation upon binding. It's a minor, but still, if you look at the fine details, actually, like some atoms are moving, like there are so-called like side chains. I mean, like it's some parts of the protein which actually moving can considerably move. So the degrees of freedom of problem is huge. And the last problem, right, that in principle, though we do know the physics, right, governing like the molecular interactions, right, it's still up to degree a model, right, because I mean like the world of proteins, it's a half quantum world, right? I mean like we do a lot of simplifications and we try to solve the problems using kind of like classical mechanics, but at every step we are sort of like having some approximations because, for example, when we deal with electrostatic interactions, right, I mean like, so in truth what happens, it's a shifts of electronegativity of molecules, so we don't have really actually full charges, right? We use, for example, so-called partial charge approximation, which is nothing more just in numerics actually to approximate quantum mechanical solution of electrostatic field of the system. So essentially like, so it's very tricky problem, right, that we try to use like a classical statistical physics, but its principle what we are just doing is we're formulating classical terms to approximate the real quantum behavior. So it's also it's tricky, and additionally it's very difficult to model. Additionally, another problem is water, right? I mean like, so all proteins, I mean like cells, I mean like they exist in water, right? So you have like a huge major factors driving protein associations, like salvation, right, which is very difficult and tricky to approximate. So so essentially if you think about it's a damning problem, right? You have huge degrees of freedom, you have sort of like your model is approximate, and then essentially you still want to get some answers which you can actually really practically check, right? And yeah, surprisingly it nevertheless is still working, which is really exciting. So how do we approach the problem, right? I mean, like, so as I said, like, to deal with such huge configurational space, right? I mean, like, what we need to do is kind of like, we're doing like this multi-scale or multi-stage approach where actually we explore our kind of like configurational landscape in steps. So first, I mean, like, since kind of like if you remember like something from statistical mechanics, right? I mean, like to calculate free energy, right, you need actually to calculate the integral over whole configurational space. So there are some parts of that configurational space which is less important than the others, but you don't know it in front, so you need to explore as much as possible. So first, we're coming up with some crude model where we can actually globally explore all configurational space of two proteins associating with each other. So it's a very simplified model, but as I will show you, it allows you to calculate the partition function of the system exactly and solve it exactly, and it seems like it's doing like some pretty good approximation. It's doing very well in actually predicting what is really important, right? And once we kind of globally explored our configurational landscape and we found the regions which are most likely the most probable for where essentially like the values of the energy is the lowest, right? There we explore it in more details using full atomic representations where we kind of turn on all the degrees of flexibility which I was talking about, right? It's a much more involved problem, but we are dealing it in much smaller regions, and so that's why it's actually become manageable. And so on each stage we are trying to come up with algorithms which allow us to most effectively to study the space, right? And if you think about like, so what we do kind of like so what is the current state of art of say protein docking, right? Essentially, like there is a competition called CAPRI. So essentially, it's a worldwide experiment where crystallographers submit your crystal structures of 2 proteins determined experimentally to some assessment committee, and it gives to all the groups in the world. I think right now there are around 80 participants groups, and essentially you need to given like the structures of individual components, predict a complex, and if you can do it right in 10 models, then you consider that you get it, right? And so this way, kind of like the current state of art, essentially level of certainty, that like given a structure, if without any additional information, we can give like up to 10 models. So of course, if you have more information, some low resolution experiments, you can actually do it better. But for the time being, that's kind of the state of art, right? So of the field. Okay, so let's go actually. So I sort of like told about difficult physics and essentially like, yeah, the challenging mathematical problems. Let's see what type of algorithms we are using. And I will try to tell you it in the form of a story. I will tell you what we sort of were using until recently. And then I will show actually you how this algorithm actually helped us to understand something more about protein-protein association, which in turn helped us to do even better and fast algorithm which we are developing right now. So in this way you can see kind of like not a static picture, but kind of science in development, that how things are moving. So the first thing what we need to do, as I told you, is we need to globally explore energy landscape of the interactions, right? So in principle, what it means that, say, for example, even let's consider like we just have like 2 protein 1, protein 2. What we need to do, we need to calculate all possible configurations of one protein with respect to each other, right? So first approximation what we do is called soft rigid body model. What we consider is that yes, it's true, some parts of the protein are moving, but we're considering the smooth is kind of is not very big. So globally we can. So what we do, like instead of like modeling the flexibility, we're just smoothing energy potentials, trying to make them more essentially to account for the things that localization of atoms is not exact, right? And then, so what we can do, if we can put like two potentials which we want to calculate on the grid, and express it in the form of sum of convolutions, then there is a very, very fast way how you can calculate that, right? And that's called fast Fourier transforms, right? So again, the idea is that like, so the simple idea how to understand it, right? It's like, for example, like electrostatic interaction, right? If you imagine you have like a potential on one grid, you have a charge on another grid. So product of potential times charge would give you electrostatic interaction, right? Between like one protein and another. But since it's a convolution, right, that for given translation, I mean, like using like Fourier theorems that essentially Fourier image of convolution is equal product of Fourier images of each individual grids. So by doing 2 FFTs to the Fourier space and back, you can actually calculate, I mean, like the convolution which naively would take you up to n to the 6 operations in n cube log n operations. So that makes it like it makes it feasible actually for the grid-based models. And obviously you would need to do like sampling of the rotational group SO3. You would need actually to do explicitly. But nevertheless, it makes it possible actually for such grid-based model to calculate full configurational integral of the system. So you can calculate full partition function given the approximation, and that's actually turns out to be like very, very useful. You would some people would argue, okay, like you're making such a big simplification, is your kind of like partition function does make any sense? But we know examples in physics, right? For example, things like Ising model, right? I mean, like it's a simple model. I mean, like which was done like which is actually based on the kind of spins in the like. So it's for modeling of spins, but it was simplified up to like 2-dimensional model, and nevertheless it still had some behavior, like for example, explaining critical phase transitions and so on, right? So in this way, so kind of we're also simplifying the model up to some really, really simple thing, but since we can solve it exactly, and we can actually check it with experiment whether it gives you the right answer, and surprisingly it gives. So which means that essentially there is something kind of like that it's a valid model, so we can validate it directly with experiment. So
how big are the grids?
Yeah. So essentially, the grids we are doing, so typical protein size is 30, 40 angstrom. So essentially, so we are using, so since we need to fit 2 proteins, so the grids essentially, so it's around 100 angstrom, and we're using 1 angstrom, so it's 100 cubes, so grids, so that's a typical size. It's a little, usually you want to pad it, right, for FFT efficiency, you want to pad it to kind of to the low product of low prime numbers, right, and things like that, right, so essentially, so what was our contribution to this area? Actually, it started back in 2006, but actually I was continuing to work it for quite a long time. That actually I was able to express a pretty realistic molecular mechanics modeling function. So essentially, all the physics which we know about the system, right, in the sum of this convolution-like expression, right. So because before what people were using just was using this FFT approach just for measuring shape matching and things like that, right. Originally, this technology was actually developed in Israel by analogy to some radar matching kind of things, right. But essentially, my work actually helped it to make it the sampling makes much more physically meaningful, because we have a really really good and meaningful energy function. So essentially, we model van der Waals, electrostatics, different forms of solvation, hydrogen bonding, and so on. So right now we have a pretty powerful mechanism that actually we can sample. We can deal with pretty complex systems, molecular systems, and pretty adequately. So the next key idea, right? So once you can sample, so essentially all possible interactions effectively, the next key idea actually is that you can really calculate now the partition function of your interaction, right? So that makes it very, very very good and nice, right? Because in majority of existing approaches for molecular simulations, right? we're running, it's usually some stochastic approaches like Monte Carlo. It's in principle, they're pretty good, but they cannot really guarantee that you explore all your space, right? So in this approach, we can guarantee that for the given problem, we can actually explore full configuration of space and calculate partition function and find actually what are the most probable regions in space which are kind of which is the most important, right? That's how you could see it, right? Imagine if you have this one-dimensional energy landscape, right? And then you have a sample points, right? So essentially, you can see that actually if we just do some cutoff at some low energy points, that actually points which these low energy points would be representing kind of this microstate funnel corresponding to a association state, and so we could actually try to kind of predict kind of what is the most likely complexes based on the probability of these funnels. So what we do next after we are done with finding the regions? Remember, it's a still crude approximation, right? we do a lot of approximations on energy and things. So right now, so what we want to do is we want to get a better quality structure. So the process is called in our kind of slang is called refinements. That you start with some crude models. And then essentially you want to get a better, more accurate model and better estimation, right, of your configurational space region using full flexibility of the proteins, right? So now we have a degrees of freedom. We still have 6 degrees of freedom, but now actually in each point we are doing local optimization, so we can move all the atoms freely. So essentially the 6 plus n optimization problem. And so how to do the search in full atomics in the space of this 6 degrees of freedom, but with a more complex energy function, which is going even beyond convolution type approximation. So what we can do, right, essentially is and this energy landscape is very, very rugged, because if you deal, for example, van der Waals forces, as you know, is proportional 1 over r to the 6, right? So you have very, very kind of energy landscape. So to do the search fast, it makes the search very difficult, right? Because if you think about Monte Carlo and everything to jump over these hoops, right? Would be very, very tricky. And so essentially, so what we do actually, we are trying to find a quadratic function which actually underestimates this very, very noisy energy landscape. And so we can formulate this as an underestimator problem. So we want to find such a quadratic function which goes below all the existing kind of data points, and we want to do it tightly. And we show that actually it can, for quadratic function, we show that it can actually be done can be formulated as a semi-definite programming problem for arbitrarily quadratic quadratic polynomial, and that allows us kind of like to have a much smoother surface on which we can do the search and optimization. So that's an approach called semi-definite underestimator. So to do this, I mean like, so okay, I mean like one thing is good is to make some algorithms and kind of develop our understanding, but also like it's also useful if people actually use it. I mean like, so to do that, actually we developed a protein docking server, which actually implements two steps. First is a global docking, and second is partition function calculation, right? And the server is called class pro. And so in this iteration, it appeared something like 5 years ago, and it's pretty simple to use. As you can see, you just pass a job name, PDB IDs, and then essentially press doc, and then essentially you can get a structure given two input structures. You can get a structure of a complex, and that it's so simple to use that sometimes actually people who even don't have any idea what they're doing, they're using it. For example, we saw that somebody was submitting, instead of PDB IDs, they use the Microsoft doc files. I don't know why, because it sounds similar, right? It's doc and DOC, right? So, but it's funny, but it happens very rare. Most of the users actually are meaningful, and since we can use, and since as you know FFTs are very fast and effective, they're trivially parallelizable. So we actually, so original version of the code, I mean like we ran it actually on the, it was back in 2008. I mean like we ran actually, it was on the Blue Gene, so at that time it was actually one in the top 50 supercomputers in the world, right? And to us it was so nice when you have an algorithm which runs like around like, I mean like maybe like on regular PC at the time it was running for like maybe for a day, right? You put essentially it on a Blue Gene, since nobody was actually using it, because for whatever reason you can do this write code in C with MPI, and people actually, at least at our university, were not really that much like writing special code for it. So what happened, essentially we had a few full Blue Gene available to us. So essentially the running time of the program was essentially, I think it was around 2 minutes, but I believe it was just the time of loading and unloading the results, because essentially it was just instantly, right? So it was so big fun, right? So, but obviously like, yeah, computers are getting faster and better. So like, so right now we actually can run it, I mean like, so we run it on regular clusters, but nevertheless, I mean like it's still pretty fast and effective. So I think like the job like runs around like for users around an hour because of the queues and things like that, right? But nevertheless, it's picked up usage. So essentially, if you look, we just checked the statistics recently. We have around more than 10,000 users. Why I don't tell it exactly, because I mean like you can use it both as registered users and unregistered users. So we have around 7,000 registered users, and in addition we have something around like 15,000 unique IPs, kind of like from each job. So we estimate that we definitely should have more than 10,000 different users for the server. And in this like last 5 years, we run around 150,000 jobs kind of for the community. And the usage is constantly growing. There are papers citing it, and we can actually already like can collect the statistics of usage, which is actually quite fun. For example, if you can see that's a daily usage of a server, so you can see there is a clearly 7-day periodicity, right? That people are not running jobs on the weekends, right? And then essentially on Wednesdays, right, is the peaks. I think it's, but I think whatever human activity you do, like I mean that would be similar type of graph, right? Yeah, so the yearly usage is kind of also funny, right? So you see like the summer, predictably people are not using stuff much, right? But interestingly, there is not the big drop off in the December, January. So I guess it's either too short or something, or people want to push towards whatever end of the year deadlines. So essentially they keep running stuff. So like, so it's kind of like, yeah. So it's interesting. And then essentially, but main thing, okay, I mean like we have the methodology, we have it kind of like that it's pretty fast, so people actually using it, right? But like, I mean, we also want to have an objective assessment actually how well it is doing. And I mentioned to you that there is this Capri competition, right? Essentially, where you're given two structures of two proteins that you need to predict the complex. And there are kind of competition like for servers and competition for the humans, right? So because humans can do whatever they want, they can run all the servers, they can essentially kind of do like all the possible analysis, read papers and stuff like that. For example, I will show you like the person who was the top performing in this competition. I'm asking, okay, how are you doing the prediction? He's saying, okay. He says, on the weekend, I'm sitting down, taking a glass of beer, and I'm looking at like hundreds of models, and then essentially I'm choosing the one which looks the best. So that's essentially the human state of art of the predictions. Obviously, servers don't have all that, right? Servers have like a pretty strict deadlines, 24 hours after the submission. So essentially what you have time is just to put the two structures in the server, press run, and then hope that the results are good, right? And so, and nevertheless, actually, for many systems, I mean, like we are getting a pretty reasonably good result. So on this color code, I think the green is the crystal structure and the blue is the model. So you see, sometimes, I mean, like we are getting a pretty accurate model. Sometimes, I mean, like not that accurate, but essentially, like in majority of the cases, I mean, like the structures actually are pretty reasonable. So how does it comes in terms of like ranking? So there is, as I told you, there is a separate competition for servers, and there are separate competition for humans, right? So ClassPro actually, it was, I mean, like pretty much all the time. I mean, like it was on the top. I mean, like it was the best server, right? So for example, in the latest round, which was in 2013, like this first number is how many models it predicted correctly. So you see it did 6 models, and the next server did 4 models, right? And then essentially, and I think right now there is something like around 15, 20 different servers and 80 groups. And this, if you can see, is the human prediction groups. So like, so as you can see, like what we are doing actually, that we are actually competing in the top 10 actually of the humans, right? So servers are catching up, right? So that's actually very good for us, right? Because as I told you, like I don't know too much of biology, so whenever I try to do things on my own, actually here you see like I didn't do like much better than the server itself, right? It was the same 6 targets, right? So maybe slightly better refinement. It's because actually, like I said, the third step actually it's still pretty computationally intensive, so we cannot afford to run it for like all these thousands of users. So, but still, it's very, very promising. So, like actually, the new Capri like will happen in a few months, right? I mean, it's still a secret what is the kind of official ranking and everything. So next time I talk, right, we'll have like a sooner date. But yes, I hope that ClassPro is still is doing pretty well. So essentially, so the point is that yes, the servers actually are not that bad as compared to the state of art in the field. And by the way, to see the difference, right? So the person like who did the best, right, is the one who drinks beer, right? So you can see like his hot dog actually he's here, so it's not in the top 10, right? So that's the difference of performance between a server and a human, right? So essentially, so there is very big gaps. So still. As that you can improve, but as that servers are catching up. So the interesting part of the story, as I told you, I mean, like we talked about this a lot of big approximations. So people say, okay, does it have any relationship? Just maybe just kind of some funny algorithm for getting you the structure of the complexes. Does it? But we do have a simplified model. We do calculate partition function. So we hope that what we do actually makes a sense, kind of it's real physics, right? And there was an interesting problem. I mean, like, so if you kind of do the sampling of protein interaction, so here what I show you is a sampling, like it's a RMSD, it's a measure of distance, it's a one-dimensional measure of how far two kind of proteins are from each other. It's a sum of difference of coordinates squared, like over square root, right? So, but essentially you can think of some form of a distance, right? So you would see that essentially if you have energy function, you have like this kind of like, you see this rugged energy scale landscape, this is a realistic complex. But you see there are essentially additional some minimas, which in addition to the main one, which is this is 5 angstrom. This is considered pretty close a crystal-like quality prediction. But you can see there are other minima. So the question is, are these minimas are kind of some artifacts of our sampling, or they have anything relationship to the reality? And until recently, there was no way to tell because there was no actually experimental techniques which actually can tell us what happens like when two proteins associate. So, but recently, like one of our collaborator at NIH, like Marius Klor, he was actually able to get a structure, get actually some information about protein-protein association using so-called PRE NMR technique. But essentially, so in essence, what it can do, like it can get you some kind of like one-dimensional graph telling you what would happen, how the protein association landscape looks like. And we have a theoretical way, given kind of configurations of our landscape, tell essentially what would be the graph, what would be that graph actually if you would consider some association landscape. And so the idea was that essentially that if to match essentially what we're getting from simulation to the experimental data. So for example, if you would think that two proteins associate and there is only near native complex, right? What you would see on the picture would be this black line. As you can see, it has a very strong deviation. So the purple dots actually what they see on the experiment. It's a pretty noisy experiment, but it's still robust to show that essentially there is some regions of space, regions are not explained. I mean that some part of the experiment is not explained using just like a specific complex, right? So there is something more. And when we plugged in actually like our encounter landscape, which we get from the simulation of the proteins, right? That's essentially what I showed you on the previous picture. We're suddenly getting like this blue line, right, which has like much better agreement to the data. I think the correlation goes from 0.4 to something like 0.8. And actually, and according to our analysis, that's kind of the limit where you can get because of the original noise of the experiment. So what was nice, I mean, like, and we did it actually not for this particular protein, but for many proteins, like there was around 5 or 6 for which there was this data available, and in all cases we got very good agreement, right? So we got to feel okay that what we are simulating actually does make sense for these systems. And so we were very excited. So when we sent this paper to review, so like reviewers who were kind of like biophysicists, they asked, okay, I mean, like you have so simple this grid-based approach. How can you tell that all this effect what you're doing is not coming from shape and not from the energy of the interaction, right? And we said, can you prove it? And we said, okay, no problem. Let's switch off all our energy, just leave the shape on, right? And what would be the picture of the curve is like this blue line. So you see it's totally off from the experiment, from the native. So it's clearly the interacting energy that what drives kind of like a formation of specific encounter complex, and so we can actually get it. But okay, so this was all just the curves, right? So let's see actually visually how does this looks, right? I mean, like, so if we kind of, all these points on that red RMSD energy plot which I showed you, if we plot it in the points, I mean, like here is the centroid of the ligand. So this is a receptor, this is a specific protein complex, and this is actually like all the configurations which you have around it. So you can see that there is one big blob kind of near the kind of correct complex, but it's all the different orientations, and there are some extensions to the left and to the right. And so this is kind of like, this is how it looks like. So essentially, so you see that, and this was actually very, very interesting to us because in kind of biophysics, there was a very big kind of problem until recently actually explaining the protein association rates. Because, I mean, like classically how you would explain, I mean, how you calculate like diffusion rates, right, essentially in like this Smolotkovsky approximation, right? You consider like the spheres, right? And then you can calculate what would be kind of like the diffusion rate of protein association. The problem is that they were too slow. So people kind of like the proteins associate much faster. People said, okay, it's all about the energy. So they actually said, okay, we can get like a pretty good approximation of the protein association rates if we assume that there is some patch around like 10 angstrom around the native complex, where if proteins somehow come together, it actually somehow magically flips in place, right? But what was the mechanism of this mystery patch wasn't very clear. And so to us, actually, it became interesting. Since we can sample the thing, we can actually look what happens to this blue patch and actually try to understand what its inner structure, and maybe it actually can explain us what's going on. So, but to do that, I mean, like we had to return a little bit back to mass, because what is the problem, right? I mean, like the space which we are working on is a space of rotations and translations, and I mean, like, okay, the rotational space is simple, right? It's just a simple Euclidean space R3, right? Rotational space is a rotational group, right? I mean, SO3, right? it's kind of like, it's a more complex construct, right? I mean, like, which is called like manifold. So something which locally behaves like Euclidean space, but globally has the curvature, right? So what we wanted to do, right? I mean, like, to do the analysis is much better to do in kind of in a simple Euclidean space, because we can have a direct meaning of what's going on and understand it, right? And luckily, like, the fact that manifolds are locally behaving like Euclidean space, right? So how it sort of like can be understood, right? I mean, like, the simple manifold, for example, is a circle, right, on a complex plane, right? That essentially you have like a parameter t, right, and then essentially e to the it, right, is a circle, right? So the connection between the manifold and the corresponding inner Euclidean space, that at each point there is a like tangent, right, is actually there is that I mean, like, if you can, so essentially like at every point you can have like a small I mean, like, you can have essentially like a tangent in a tangent space. You can have so your Euclidean space locally, it's actually can be considered as a tangent space, and then you can actually have a very nice canonical projection from this tangent space to your manifold, like, called exponential map. So essentially, so what is the idea is, like, so for example, I said on the circle, right, if you have parameter t, then e to the it will give you parameterization on the circle, right? So the same thing can be done actually also for the sphere, right? I mean, like, for the sphere, the tangent line is actually is a plane, is R2, right? And again, there is some generalization of the exponential map, which gets you like parameters, so like on the move the plane, right, to the sphere, right? And so this is very nice, and like, yeah, so essentially if you're familiar with Lie groups, right, that essentially like this manifold, right, is called like I mean, like, for example, sphere is a Lie group, right, and this projection is it's algebra, right? So like, but essentially the point is that we can have like locally, and since we are dealing with locality, right, you can have locally mapping from the Euclidean space to the manifold, and that was very nice, right, because we could actually convert, so we can actually analyze our encounter landscape, actually not in the manifold, but actually on the Euclidean space. And that it makes it very, very, very nice and effective, right? Because we could actually understand what is the inner structure of the space, right? So to do that, right, I mean, like, so once you have Euclidean space, right, I mean, like, you can do PCA, right, since there is no kind of like dependence on the coordinates and everything, right, all the coordinates are independent, the PCA will give you like an inner dimensionality of the configurations, right, and geometric meaning of it would be actually hyperplanes which are bounding the region of interest, right? So what we are doing, right, is we took all the points which we have, and they are shown in blue, so in light blue. And then we took a low energy points. So essentially all the points which kind of like are like the lowest energy. And they are shown in dark blue. So like what you could see, that essentially when you do like the sampling, you see is exhaustive, right? So in all degrees of freedom, we're actually looking not 6, but 5, because we're looking at the contacts of the proteins, so two proteins coming together. So you would see that actually that you will have like a sampling on all over the space. But if you look at the low energy regions, they have only like 3 degrees of freedom out of 5. So essentially what happens, I mean, like energy function makes like essentially a low energy subspace, which is like where all the points are located, which essentially makes on this 5-dimensional space a smaller dimensional canyon, which actually can accelerate protein association, right? So that essentially explains this paradox, right, which we are talking about, right, how when proteins come to this big patch, how actually it's kind of like it associates so quickly. So what happens, there is a lower dimensional pathway which actually puts the proteins back into the place, right, and it's purely driven by the energy of the interaction. So essentially, so we have kind of like a geometric explanation, right, of the effects of energy function on the protein association. So to see kind of how these things look like, right, it's in principle, right, it should be like some motions, should be some combination of translations and rotations. So you can see, so for example, for this protein association, it's a pretty flat interface. So you see this purple actually is a native structure, and you see the motion is some form of a point rotation around the contact point back over there. So you see like two proteins are moving. This is a permitted, the permissive direction, so the corresponding to the largest eigenvalues of PCA. And this is actually the restrictive direction. This is a translation. So protein cannot really actually translate this way. So essentially the idea is that when the association in this case happens, so two proteins come together and they get to the lock point and then they can actually reorient very quickly. So we did even some small movie. It's not really dynamics. It just the model is that it's implicit solvent. It's implicit solvent model. So we do account for solvent, but implicitly. So essentially, like you can see actually how the this is as it's not a dynamics. It's actually just a statistical picture from the statistical ensemble. But nevertheless, you can see what type of motion it's going on. So okay, so this was very cool. Was very cool and all, right? But essentially, that gives us a very nice understanding of what's going on, right? So now the question is, can we actually use this understanding for designing a better algorithm? Because you see there are a lot of things. So it seems like it's very effective to consider the interaction of proteins actually like not on the separation of on translational rotational space, but on the manifolds, right? That's number one. And second, energies restrict the dimensions, right? So the question is, can we actually use all that, right? And get this and make it work for a better algorithm. So the first big idea was that, okay, maybe actually instead of like separating the rotation, so like rotational and translational space, let's look at the generalization on the of the FFT to the manifolds, right? Because in principle for SO3 group, you can actually build a generalization of the manifold, and this will allow you to calculate the interactions. It will allow you to calculate like the interactions much more faster, because like again the idea is that instead of like looking 3 translations and 3 rotations, right? What you can have like 3 rotations of the ligand, 2 rotations of the receptor, and just translation along the contact point. So you can actually have like a representation of a configurational space as a space of actually SO3 as a square of SO3, right? I mean factored by 1s, right? Because there is a redundancy of rotation of 2 bodies with respect to each other. And then essentially, what is interesting turns out to be that the key like idea of like FFT, right, is essentially that operator of translation in the exponential basis is just multiplication by exponent, right? That explains this is main part of the FFT convolution theorem. Turns out to be that if in Euler angles, actually the expression of the rotational operator, and that's actually like where the quantum mechanics comes in, right? Because you can see these terms, right? It's called very well known like Wigner D functions, right? They look exactly like actually in a very similar way as translation in the space, apart from that we have like in 1 degree of freedom, we have like a small d, right? Which accounts for the curvature of the sphere. But the structure of the expression and everything is very similar to actually Cartesian FFT. So what we were able to do, we were able to formulate, we were able to generalize actually FFT to the manifolds. Actually, like it's not just our work. There was actually several groups who formulated like FFTs for SO3. So we use that work actually for our SO3 square. But the nice part, we can do the search now not in this artificial separations of rotation and translation, but directly in the space of this encounter complexes as we discussed, right? And if you look actually at the search, so we did implement this algorithm. I'll show you the formulas in a second, but you can see actually how the search is performed. Sorry, 1 second. How the search is performed. You see, it's kind of the search is actually now is similar to actually how like the encounter complex is associated, because that we do it in the proper space. You see, like actually the protein was at some point coming to the right thing, but then essentially exploring all the other configurations. So essentially this is like much more effective way of doing it, and we're actually achieving around 10 times speed up, as compared to the Cartesian representation, and the accuracy is very comparable, right? So that's essentially the generic expression of our FFT, right? So you see it has like FFT, like real FFT-like terms, but also it has this two kind of like D polynomials, right? Which actually at this point we calculate, we have this quasi-fast manifold FFT where we accelerate the calculation of this expression using FFTs, but these ones we're actually doing explicitly. But actually there are ways to make it even faster using discrete Wigner transforms. So, but essentially the essence, the idea is absolutely the same. What we did is just we realized that we're working on the manifold and we generalized the formulation from the classical FFT to the manifold. And this has turned out to be very powerful since it's much faster. We can do like much more interesting things. Like for example, we can deal with flexible, like say NMR ensembles when you have like not just a protein with protein, but many proteins with many proteins. So it becomes very, very, so we can do it like, I mean, like things much faster. Also another advantage, actually, we could have much more complex energy functions because the way how we formulate the expression, we can actually get the summation of convolutions, which was before need to be done for each energy term separately. We can do inside the loop, so we can actually have very complex energy functions like experimental restraints and so on. So it actually opens up some great avenues for speed up and applications. Okay, so that was one idea, just realizing that we work on the manifold to do things faster. So the second idea for the underestimator, right, we worked in the 6-dimensional space. I mean, like, and now actually we realize that we can actually cut this down like to 3, right? Because physics shows that actually that's where the search is going on. And we already kind of pre-sampled the region, so we know what is the key region, and we know that they kind of agree more or less in the representation, so we can now do underestimation in lower degrees of freedom, which makes the algorithm faster. And generally, manifold representation, actually, you remember there is also a step of this local minimizations where we can have like approximations this rigid body with some kind of flexible movable parts, right? So we can have like actually by selecting largest rigid body of in the interacting proteins and actually moving it, optimizing it on the manifold, and with other degrees of with other flexible degrees of freedom, you can actually save a lot of time too, right? So we showed that we can actually get like just going to the manifold, we can get up to 10 times faster approach. So like you can see like how like the minimizations kind of like happen. Like for example, this is kind of like a minimization in the some experimental data called SAXA, small angle X-ray scattering. So you can see that so we can have like when you minimize two proteins when considering this one as rigid body, but using full atomic interactions plus this scattering data, we can very quickly put it into the place. And the same we can do also with flexible molecules. So you can see actually like, yeah, it's again like the largest part was considered rigid body, and then the rest is a flexible internal coordinates. So as you can see, we get a lot of mileage from the idea. So that was kind of like one of the key messages which I wanted to show you, right? That essentially that given essentially like a pretty simplified model, but at the same time being able to solve it exactly, it allows you actually not just kind of solve some practical problem, but also get some insight in the physics of what's going on, and that physics actually and this insight actually helps you to actually design even better and faster algorithm, which we can do, right? So I think like, yes, there is remaining like maybe like 5-7 minutes. I mean, like I will just briefly skim through kind of like a couple of other applications of these techniques to show that it's not just proteins, and then I would be happy to take the questions. So we did. I mean, like so some of the key, some of the cool applications what we are doing with it right now, right, is actually doing modeling not just as a protein association, but actually some not just complexes, but actually trying to get some physics from the interaction. So we look at the three. At several classical problems which are currently a very challenging kind of computational biology. So one problem is the following. When you get a crystal structure of a protein, right, you need actually to know whether it's crystal, whether it's a realistic kind of biological interface, right, or it's an artifact of crystallization. So this is a very long classical problem because actually crystallographers want to say that everything what they get is real, right? I mean, like, so biologists say, okay, we don't fully believe it, right? I mean, like, so some of them is not, right? So another problem which is also very interesting is aggregation. So when essentially two proteins, when protein in presence of some other proteins or together, I mean, like sometimes protein behave like not normally that they behave in solution, but they come together in some globules, right? And this is very important, for example, for antibody, like you know, like for example, antibodies, right? It's a 50% of our existing drugs. and for example, and essentially, like when people actually get a good antibody, they suddenly see that it starts aggregating, and it's a big problem. So they want actually to have methodologies to tell in advance whether the thing is kind of like is real, whether there is an predict aggregation in advance, right? And finally, there is, as I told you, there is this big progress in sequencing, and people actually can get a lot of sequences. But what they want to know, actually, for example, when design vaccines for HIV or flu, what happens? I mean, like they want to find actually the sequences of antibodies which is most strongest binding to the antigen, right, and to design a better drugs. And so all these problems actually could be now targeted, I mean, like with this type of approach, right? So like one with the dimer, right? I mean, like the idea is very simple. Since crystal actually, I mean, like, so if the crystal, this is, for example, examples, right? This is a real crystal, but it's actually very small interface one, because the current existing method called PISA, essentially what it does, it just calculates the surface area of the interaction, and it then essentially decides based on it, and then some kind of knowledge-based like machine learning things that which is trying to say, okay, if it's realistic or not, right? And then this is kind of, and this case is very difficult for this PISA because, I mean, like it cannot really discriminate it correctly because it's so small. And then here there is a huge interface, which is again PISA says that it's a real dimer because it has a huge area, but it's not physical. As you see, you can even look at it; it looks a little bit messy. But of course, maybe it's a post factum. That's how human mind works; that it tries to do it. So essentially, so what we did, I mean, like, so this set, I mean, the original set, which was consisting around 100 proteins, which are real dimers, but which PISA has trouble to discriminate. And the other one, the false dimers, which actually, again, PISA has trouble to discriminate. So essentially, it's all these 100 systems where PISA is not working. So by looking at just what we do, we just simulate the association of two proteins and see if there is a high probability region near the crystal structure. And if it is, then we say, okay, it's a real thing, right? And you see there is no kind of learning, there is nothing, we have only one parameter, right? The like probability of the funnel, right? So, and then essentially on this PISA tricky case, I mean, like we are getting actually very, very good discrimination. So we can actually tell in majority of the cases whether it's a real dimer or not. And like, I think like our F1 score, like this true positive versus false positive rate is around 0.9, right? And so what we to do actually, so right now how the PDB deposition works, that actually people deposit their structures and PISA predicts whether it's a dimer or not. And so what we showed that actually, like what we did, we repeated this experiment, like it's now not on these tricky cases for PISA, it's on normal cases which crystallographers deposited, right? So in the last year. And then turns out to be, and then we dig back the literature and found whether people reported as a real crystal or not. And as you can see, like majority cases, PISA, it's actually, all the methods actually, like there are total 50 proteins. So all the methods are not perfect. I mean, like there is, so PISA gets 32 correctly, EPIC gets 23 correctly, and actually our approach gets 36 cases correctly. Actually, if you join those predictions, we can get like saying 2 out of 3 is correct, right? We can get actually up to like 41 cases right. So what we're actually thinking about that we are in contact with PDBs that actually, since we developed so fast methods for kind of for doing like this interaction, it seems like it's working like, I mean adding this approach could be much more effective. So we could use actually it for validation of the crystal structure. So this, which is very cool and exciting, right? So another application is aggregation. So again, so here is an example of a wild-type protein which is not aggregating. And so what our experimental collaborator did, he actually introduced some mutations and saw whether it causes aggregation or not. So here is an example, kind of like when actually introduced mutation is causing the aggregation. So you see the wild-type, all the encounter complexes. So these proteins are not interacting with anything. They are on their own. So we're looking at very, very subtle effects, the non-specific self-interactions. And as you can see, that like that specific complex is that essentially that introducing the mutation creates a huge encounter complex near the mutant region. And this guy turns to be aggregating. So for non-aggregating case, you see when you introduce the mutant, both wild type and the mutant is not having anything in the region. So we really can see like by looking at the encounter landscapes, if the suggested changes actually is creating like some like funnel, I mean like encounter funnels in the sync, right? And finally, yeah, and this is again has very good discrimination on our set. I mean, like this methodology works pretty well. And so finally, for antibody maturation, essentially like where we have like a huge number of sequences, and when in this case experimentalist actually measures the affinity. So by comparing actually like now we're actually going from a pairwise interaction, we're comparing different interactions, and we could actually still by calculating partition functions and comparing them, we actually can have a pretty good discrimination of affinity. So the correlation is still not perfect because antibodies they have a big flexible parts like loops. So actually we can with the faster method we could actually account for that, and hopefully we can improve it even further. But even in this shape is already useful. So I guess, yeah, this is time is kind of like is almost done. So I want to leave some time for the questions. So like, yeah, you're welcome to ask if you have. Yeah, and thank you very much for the attention. Yes.
So the brain of your model is shown in one transparency somewhere after third of all which are the interactions, actually the energy of
Yeah, yes.
Can you go there? Yeah. So this sounds to a pretty restricted set of interactions because I don't see any stacking or cross-stacking, not hydrogen bonding.
No, we do have, as you see, lower, essentially that was our original energy function 2006, but actually we added this term, so we now can have combination of those.
This is not the full list of the terms.
No, this kind of, this is a list, of the terms, right? You see there is hydrogen bonding here, for example, right? We don't have pi. No, there is no pi, right? I mean, like in all force fields, actually, we are copying molecular mechanics force fields, and most of them, even like full atomic, like this Newtonian force fields, they don't have pi stacking. Because the problem, what happens actually, if they somehow they cannot kind of calibrate it towards the other terms, so it causes troubles. Right? Yeah. Okay, so there are several ways of calibration. So like originally, like how when all this field started, so people had some, in force fields, they had some simple experiments, right? I mean, like some kind of like solvation experiments, like water to octanol transfer and everything, and they kind of match the parameters the way that simulations would reproduce the experimental data. So lately what happened essentially for this big macromolecular applications, people figured out that actually it's not possible to have like a universal energy term for all possible application. So you just write essentially like all the energy terms which are kind of relevant, and then essentially you have like a small subset, original subset of data where you can try again to match the data to match the experiment, right? For example, to reproduce, say, like for example in protein association, you want to have that you have like a lot of like near native complexes versus the others, I mean, like and so on. But this is done on a restricted subset. And then the model still seems to be transferable for like a very broad application. So we usually fit it like on say like 5-10 complexes, but we run it on hundreds and thousands, and it still makes sense, right?
Okay, like it is done in building of molecular potentials.
Yeah. So
then what is the coupling of with solvent?
So how we deal with solvent? Yeah.
Between equation or?
Yeah. So solvent, I mean, like there are several ways. I mean, like so one way for the simplest form of solvent, which is kind of the most kind of effective, is the so-called Miyazawa-Jerningen type potentials. It's inverse Boltzmann potential. So the idea is the following: you observe how kind of your system behaves, kind of like, I mean, like so what interactions happens in reality, and you assume that its probability distribution is proportional to the exponent of energy. So from the observed probability distribution, you devise what you should be the energy terms. And so in this formulation, you can actually account for solvent implicitly, because you think that, okay, you interact either with atom-atom interactions or atom-water interactions.
So you have Brownian term.
Sorry?
You have a random Brownian term.
It's not really Brownian. I mean, like I said, it's so-called like as I said, it just so in the simulation, the water it's called implicit accounting for it. So the probability of atom. I mean, maybe we can talk offline. I can show you the relevant papers on this, how this works. So like, yeah.
I have a question. You
So again, in this model, it's called implicit methods for solvation, right? So you know there are the simplest model of solvation is SA term, right? Essentially, the surface tension-like expression, right, which tells you that essentially that the water solvation is proportional to the surface area which you remove from the water, right? The slightly more advanced set, it could be like this, is a so-called like this Miyazawa-Jorningen type potential, where you have the several different atom types, including the water. And then essentially what you can see when you look at the realistic molecular configuration, you can observe what is the frequency of contacts of atom contacts, right? And whenever there is an empty space, you consider it's an atom-water interaction. So you can calculate frequencies of all these terms, right? And then you assume that essentially these frequencies is according to Boltzmann principle, is proportional to the exponent of the energy. So by knowing probabilities, you can derive interaction potential energies, and then and you can get a water term. But then the water term kind of accounts implicitly, because essentially whenever there is no water, whenever there is a real interaction, there is water. I can again, I think it's the same, it relates to the question. So I can show you there is this classical set, like 85 paper, like on Miyazawa-Jorningen and inverse Boltzmann potentials, right?
So I was surprised at the success you had with systematic underestimator, or semi-definite underestimator, which is a great name. I mean, so you had, I think one of your early slides was basically showing a very rough energy surface.
Yes.
Right. So my intuition would be that your underestimator would just be a flat line.
That's actually, yeah, no, that's
so I was wrong. So what physics is making it?
I mean, yeah, okay, no, it's the forces which are kind of like more longer range, yeah, that's true. Like, yes, because it's noise, it's van der Waals what gives you, right? If you look actually at solvation and electrostatics, right, I mean, like those actually are longer range. And so they create kind of the smoothness of the funnel, right? Because you saw like this, you have in this PCA analysis of the encounter complex, right? That's the place where the most dense region where we work, right? That's actually those two forces which are acting, right? And they're definitely creating like this smaller subspace. So clearly there is so that's what they do. I mean, so that's these two forces which actually like, yeah.
This is now a complete technical question. So you've obviously got some very rough, very high dimensional things to look at. Have you found any good visualization tools or techniques?
That's a challenging thing. It's all the time kind of difficult. Yeah, the short answer is no, right? I mean, like we are trying, I mean, we try to play, right? I mean, like with the things, but it's, yeah. So the truth is actually most of the stuff what we do is on this like S3 manifold, right? Essentially the like rotational translation or a group of affine whatever transformations, right? That's our kind of key space where we work in, right? I mean, the higher dimensions, I mean, is that what we do, we locally minimize to a point on that space. That's so far our approximation, right, which works pretty well, right? So, okay.
Very good. Thank you very much.
Yeah.